Maintaining
That's what you get.
I'm pretty low.
Yeah.
That looks like it could be parantopathes. That is less than 10 centimeters, probably. Gary, any thoughts on that? That is on our list to collect.
Let's get a little less light, a little more focus.
Yeah, hi Danny, you might be right.
Might have
It's
to zoom out a touch, Wayne.
probably a good thing to take.
Coming wide.
Just a touch till it's back in the video.
Sorry.
Gary, can you say that again? There was a talk over that.
Yeah, Danny, I think it's, you're right, it's probably what we're looking for, so you might as well take it.
Okay, are we able to collect that?
Yeah, I think so.
And what was the name of this coral?
This is potentially parantopathes. Parantipathes.
Okay, great.
This is a black coral, and we're interested in it because we've seen it before, and it can sometimes be confused with other black coral species.
No, I'm moving east. Get to you.
We're moving the ship east a little bit, guys, to give us some slack to. I can try the straight in one. Poor suction sampler.
Okay, we're almost there.
It's pretty steep to be straight in. No.
Okay, I think I've stopped moving.
You haven't stopped moving.
Sorry?
You haven't stopped moving, but you'll be getting closer.
Oh, gotcha.
And
Need
again,
to get you
sample
up to speed on
collections
this
are
thing so I can balance and you can grab.
biological
good.
sample
Both of you.
collections are really interesting because you get to see Herc's arm doing some pretty fine maneuvering. And these samples are shared with local scientists of interest in them, and then all of our biological samples are housed at Harvard
Pan right with
University's
bubble just a hair for
Museum
me.
of
Show me in a
Comparative
minute.
Zoology.
You can zoom in there a bit if you want, Wayne. I'll center it up for you.
Yeah, that'll help. Thanks. All right, zooming in. Oh, nice shot. There we go.
That's a great shot, thank you. We get, I guess lasers are off screen, huh?
You want to chase the tip of the minute there?
Okay. Coming wide.
That's good there.
Oh, yeah, I'll stay partial wide. There.
I like it when the jaws come into the shot. They sneak up on you.
Wasn't expecting that.
Look. Since it's small, we can get the whole thing.
Okay. Get the manipulator parallel to the cliff here without being spastic. Totally being spastic.
That's pretty good. Coming wide. Oh, well.
I'll bring it right to you.
All right.
Put it up in the light a little.
Yeah. All right. Oh, that's good. That's good. Let me get the focus. Stand by. Yeah, that's it. That's in focus.
Is this going to be C, is it?
Yeah, we could put that in starboard C.
Yeah.
C?
Okay.
C, correct.
Raja. C. Okay.
All right. Coming wide.
Raja.
And
You
that
guys
is
happy with the
sample
shot there? We can
010.
put it away.
Full wide. And set 2 is starboard. And set 3 is starboard. All right.
Antipytharians are usually floaty, so you might want to do the vertical again.
Yeah, especially since they're not too jammy. But yeah, let's do it. Hey, look, there's Bobby Argus.
Ready?
Yeah.
Perfect. Okay. Ready for it to come out?
Should we disable before we come out for the other samples? But can't hurt.
Your call. Okay.
Yeah, go
Ready?
ahead.
Thruster secure. And trays coming out.
What's happening here?
Your
Come on,
laterals are raging.
Yeah, I see that. I think I'll come off before we Can you disable that lateral too?
I don't. Probably.
Oh, look at that.
Lat, yeah, it'll be both lats.
Yeah.
Yeah, do it. See what happens.
Okay.
Oh.
Lateral
not good.
thruster secure.
I'm just gonna kill everything here and float. We won't go far. You're watching us in Argus, right?
Yeah. Right.
Okay, retract.
That came out of somewhere.
Okay, stop. Stop. Your other floaty bits are escaping.
Yeah, it's possible to try and scooch those back into the box.
What is...
Antonella, you're going to need to start coming up.
Sorry?
You're going to need to start coming up. 5
Roger.
meter delta.
Thanks.
Really?
I don't think it's there anymore.
No.
Is it possible to turn down the gain on that camera?
No. It is what it is.
Really?
I can't tell if it's caught in there or if that's a tie wrap. retract.
Oh, there it goes.
Right.
Oh,
You can close the box.
Oh. Okay.
Sorry guys, so we're floating up the whole time, trying to
Thrusters re-enabled.
keep that with our thrusters turned off, and didn't work out too well. I'm good to come back down now.
I think we still had some pieces in there.
Go ahead. Yeah, I was worried about, so the other one is a floaty bit too. It was floating up under the lid of the box. Not 100% sure it's in there.
Danny, one of our viewers is curious, how many different types of animals are on our collection list?
Yeah.
Wanna
That's
show
a
me
good
the
question. I
map
haven't
again?
counted them up, but I would say we have, I don't know, maybe 50 or so species at least on there. Maybe 60. Okay, I'm going to give, thank you for that collection. That seems like a good time to pass it on to our next watch. So I'm going to hand this off to Jan, tell her what our next goals are.
Great. Thanks everyone for tuning in. We're about to go through a watch transition. So just hold on tight, and your next group of Exploration Corps will be on board soon to continue on with exploring Box Canyon. Thanks so much for tuning in.
Hey, Greg. Yeah, thank you. What's your heading when you square up on this thing? Interesting. Okay. Because I want to go 035. Doable? We can just hit the slope. We can hit it as it is if you want. Oh, I was wondering what was going on. I'm sitting here talking to SPL by myself.
Hi, Rennie.
Hi.
Hi, Rennie.
Hey, everyone.
You're not alone.
Oh, thanks, everyone. Okay. So is the plan to head up to 2E? Oh, they're pouring over Sanskrit.
It is the plan.
Thanks, Jamie.
I'll just wait till. See all my snacks?
Yeah. Look up with Bubble and see if we can see if the porch light's on. Just look sideways that way. That looks like the porch light's on. It's the only light inside that's pointing down at us. So let's go here, and I want to see if somebody figured it out or whether it's ganged up with these. It's not ganged up with those. It's not those ones. Okay. It looks like porch light's on, right?
Kind of does.
Yeah.
Did you talk to them about that?
No, I forgot.
Do you want me to zoom in on Argus?
Not
Would that
really. I
help?
don't think that's
Could
gonna
you
help.
see things?
No.
Oh,
I know. Sorry.
okay.
She's itching to do something.
Yeah, check to see if they checked when they last checked the numbers, and then. Okay, so then yeah, we'll do it.
It does look like porch lights on. You can see the shadow.
Yeah, we'll do a new round of numbers.
But like,
But
we don't want that on in this kind of viz.
I know, that's what I'm. Roman Hotel?
Hey.
Found it. Porch light is Roman Hotel. We're
Good
gonna
job,
write that
Greg.
down.
Soylent Green is
in
people. Porch light is Roman Hotel.
we should write that one down and change that.
Nav, am I correct that we're waiting for ship move right now to
Negative.
catch up? No, we haven't started yet?
We have not started.
Okay, let me get the scientists to get that moving then. 1 sec.
Yeah, we just want to confirm before we go.
Yeah.
In the meantime, I can go back to.
What are we looking at?
Getting rid of mud.
Oh. All right.
Okay, so the scientists, before we start moving, would like to take a peek into the forward bio box to see how much of one of those corals remains in there, if any, so that we know whether we might want to grab another of that same species if we see it. So we just want to open up the tray and look down and see what's in there.
Okay.
That one's going to take a long time. It's so caked it might just stay like that for a bit.
Yeah.
Kick it off with Mongo.
I thought about just trying to use the scoop and scraping it off.
Yeah.
Okay. Seems pretty solid.
Yeah. Now you need Roman Hotel back on.
There we go. That one sounded like it worked.
Over.
All right. So Brandy, right? Let's see. Can she hear me?
We're just gonna get bubble on it here in a moment.
Brandy is ready.
All right.
What's that?
We're gonna
look
check
back
one of our
at these two. Can you say it was in the forward?
Correct.
Okay. So
Is
yeah,
anything
just spin
floaty?
around and look back in.
It might be floaty. Some of it already floated out, but
Okay.
we need to know what's in there or not in there.
And we're
so.
looking in these two, Lambda or Omega?
Yeah.
Okay.
Might be floaty.
Okay, well that's some definitely still in there. It doesn't look like it's floating out, so let's see.
Yeah, that looks like a decent amount.
No, I think that's...
Can we keep going out a little bit more? We're interested to see. Oh yeah, that's plenty in there.
Oh, there's plenty in there, yeah.
I think it must have been a small piece that got lost then.
Yeah, and it looks like we almost have that base part too.
I think you've got at least a partial base, so I
we're
would
good
say
on that.
I'd say we're good there. Yeah.
Okay.
Okay, I'm just going to reclose this so that we'll pinch that one branch.
This is Danny. I think it was the other drawer.
Oh.
Oh, the other drawer is pretty empty.
Was there anything in Omega on your sheets back there, Jamie?
Maybe she means the starboard bio box. There's nothing in Omega.
No, not Omega. The other
The side
The other
drawer.
side.
drawer. Yeah.
Well then, I got incorrect information in the handoff, and I don't know which one might have floated away.
Okay. So
what's Jamie? What specimen number is in Lambda?
I called them A and B, so I'm a little confused right now. This is new
Are we
to
okay
me.
to move towards 2E?
the reason that we stopped calling those two A and B is because there's an A and B on the starboard bio box.
Yeah. Okay. So which one's which?
Lambda is on the left. Omega's on the right.
Correct.
Let's give Jamie a little bit. Time
Nothing
here.
else was in the forward bio box. It
Okay.
was just the left side.
Are we good to move?
Yeah, we're good to move now.
Okay.
Let's move on towards 2E.
Got it. Let's go 015 for now, just because the slope's a bit
015.
more towards that, and then we'll eventually be heading over a little more eastward. Bridge nav. I think we could do
So,
30 because you're pretty far.
what do you-
Argus is pretty far.
Yeah, I
away.
was going to ask you to swing over.
Yeah. Could we have a step 30 meters, bearing 015? Thank you. There is that though. Nah, that's good enough. That's 40 away.
Yeah.
So what floated out was in a drawer that was by the push cores, or what looked like handles for the push cores.
Gotcha. So starboard bio box around the side.
Was it the big containers over there?
Back row, did you hear Danny?
Yes, but I still don't know which
Right.
one it is from there. We can't really look
Do they
in
both
those.
have coral?
So
Is there multiple corals back there?
there are,
Yeah, and we
Yeah.
can't really
and
look
we
at
can't
any,
look
right?
in there anyway. So it doesn't really matter at this point. We should assume that we don't have a good enough sample of whichever one it is, which will need to be confirmed by people in the lounge.
Yeah.
From the last
it's still
watch.
That cry noise is
unchanged.
hanging out.
7. No, that's the one that we thought was in question that's no longer in question. Yeah.
Ooh, color change.
Yeah.
Sorry.
Okay, got a ship move coming in. So he's moving you, what,
015. So we're going to want to shift over to the left. Yeah, that way, and then Argus will change its heading. to around
All right.
015.
So just kind of follow me around until you get to 015 on your heading. So it's the minus, right? Because your heading is what, 60? Yeah. Don't use those. Since you're in auto head, you can use the buttons. Yeah. That's the end goal, but we want to keep me in frame as we go around.
I kind of want to just not do another move after that. What do you think?
Like not move that way anymore? Or?
I could do another one, but we got this little unknown wall if we think we can get above it quick enough.
Oh, we can come up with Argus or do it.
All right,
I'm still about 20 meters up
I'll
front.
add another. Bridge nav. One more step. Thank you.
Let's go ahead and stay stretched out like this.
Ooh, that's cool.
That is a piece of something, I think. Not an actual something. Oh, those are the special snacks. I know about everything. I do know where those came from. Video, let's go for a zoom.
Yeah, let's see if there's anybody hiding in there.
There's something black inside.
Maybe it's a gopher.
Maybe.
Because you did say it was a gopher zoom. I don't know if everybody heard that.
Oh yeah, I did hear that. Maybe it was just the shadow of the holes.
Yeah, I think it's actually the base of the sponge.
A couple more weeks of summer then, I think.
Not according to the weather.
Well, it's
the weather is in
on the ground, so does it make it a groundhog?
ground sponge?
I don't see anything in there.
No, I don't think anybody's home. Just on the outside.
Would you call this a barrel-shaped sponge?
No. I would call it a Picasso-shaped sponge. It's not as ornate. We might want to collect this.
How might is might? Because I would have to stop this ship move.
Danny, how might is might? Because the ship has already started.
Yeah, I can stop.
Well, we could keep looking for it, but looks like one of the morphologies we're interested in.
Seems like we should sample.
Yeah, let's go ahead and sample.
Bridge nav.
And if we could,
Hold position.
yeah, get the edge, the lip of it.
Okay, sampling and going for the lip.
How do you think about sampling about this thing? It's on the kind of on a wall.
Well, we'll see what we can do.
It's gonna almost disintegrate, I'm afraid, if we use the slurp. So is it possible to try and cut a piece of the lip off? Like maybe a wedge-shaped piece?
The problem will be storing a floaty piece of sponge.
Yes, it will.
Mid-water, because there's no, I don't know if we can land.
But you guys are the best pilots in the entire world.
You can do it.
You can do it.
All right, Aaron, let's come full wide.
Okay.
Figure out what we need to do.
We could do a soft slurp.
We can control the speed of the slurp, and so we could do a soft slurp.
but maybe we could try the grab first.
I
There's
don't
plenty of
know
sponge.
if the slurp is functioning. I heard rumor hours ago that there was something stuck in it.
Huh.
It wasn't part of handover, though.
Could
So
clean it out with a sponge.
Yes. Thank you, Rennie.
Well, it is a glass sponge, so it will be pretty harsh. These are not the type of sponges that are like loofah sponges that you're going to use on your back.
It'll be good for scrubbing. You know, lots of abrasives.
I'm just going to watch Argus's mezzo.
Yep.
So, Paul, that's what I'm looking at up here. I just want to make sure I'm not driving Argus there into that wall.
Yeah.
We did just complete the ship move, but there'll be a bit of swing. I'd like to stay 20 meters away, so I don't really want that any of those sonar hits creeping in.
And we're probably at about 25 right now, so close.
Yeah, yeah. It's not like the worst. 20 is still a bit of a buffer, but I might back it off then.
And what would you do in that case? Call a ship move?
Yeah, I'd just go reciprocal of the last one I called in. And I cheat by looking at this compass here, and I just go the opposite, so 195.
You're not allowed to give out your secrets, Rennie.
Oh, yeah. I mean, I just have it all. I have every reciprocal heading in my head.
Wow.
And you still have time for jokes.
Nice, Greg.
Oh, little bivalve swimming around. Not too much.
Flying bivalve.
So it's about 20 centimeters across and maybe 60 centimeters tall. That's a big one.
Rennie, you want to hit stick lock?
Yeah. Ready?
Yep.
Yeah. Dr. Henry Reisberg will be looking at the spicules and the pieces of the sponge that are kind of along the lip, and that's the taxonomy. That's the main part of the sponge where he likes to use for taxonomy. And he has to put
Jamie,
everything
what
under
box
an
is this
electron
going to go
microscope.
into?
Assuming that you pull it off and don't slurp it.
Yeah.
It would probably be good to put it in one of the forward ones since it's going to be floaty.
Can it go in with the
yeah, Jan,
coral?
are you okay with it joining the coral, or would you prefer it go in the empty box?
I think that with the coral.
Okay.
Because
We'll
that way that drawer is going to be, and the coral might actually help keep it in there.
Yeah, we'll just keep an eye on if it has a brittle star associated or not on the piece that gets pulled off. Make a note of that.
Were there any associates on the other coral?
I don't know.
Let's see.
Stay.
None were noted.
Oh, before we collect, let's
Yeah.
make certain that we're getting full photos. You got full photos, Jamie?
Yeah, I've got photos of the whole thing. I don't know if you want another zoom now that they're a little bit more stable, but we got some good zooms before too.
Let's get a good zoom of the lip.
Short video, let's push in.
Stability zoom, aye.
And if we could get kind of like a background that's a little bit darker than the sponge itself, like, there you go, excellent. And are lasers just no longer available?
No, they're on. We can't see them when we zoom
out
this
of screen
close.
when
Okay.
we zoom in that far.
I do have some frame grabs with them though.
Okay, that's good.
Okay.
All right,
I'm good on the grabs.
I think we're good.
Okay.
We're
All
good,
right,
Jamie?
Aaron,
Yep.
Okay,
let's back out.
that's an awesome photo too.
Yeah, with the crinoid just hanging out.
Yeah.
Ooh, jet pump's not super heavy.
No. 29 knots. Can we get to 30?
How about we not play that game? All right, Paul, if you'll follow the claw on bubble.
Yeah.
So we want to get both sides of the claw in frame. There we go. Just gives me a slightly different perspective.
Do I have a zoom out? No.
No, no zoom out, but we can do... Bernie, are you happy? Argus seems pretty stable.
Yeah.
Okay. We can do that.
Got it.
It's just not enough.
Somebody ejected an arm over that. Nice.
It's tougher than I thought it was going to be. Oh, very nice.
Well done.
Definitely have associates. Do you want me to shake it and lose some associates, or?
No, they can stay.
Okay. Video, let's push in. Oh, where are we? Roman Hotel.
What's R in military, like comms?
Romeo.
Romeo. So if only it was Roman. Roman Hotel.
All right, and we're going in with the coral. Jamie, are you happy with this? Okay.
Yeah, that's good.
All right. Aaron, let's come full wide.
So that's lambda.
Lambda.
And we're going to go. Into here.
That means you guys got to find the special character when you print out the next set of sheets back there.
It does mean that.
Oh, yeah, yeah.
I labeled 4 bio boxes one time. Chandler, Monica, Rachel, and Ross.
You're not going to confuse those. Camera's back. So when we're doing stuff this close to the camera, we always make, especially with these jaws, because they go so wide, we make sure the camera's back and just are going to be careful as we let go. All right, nothing floaty.
Oh, I forgot you could do that on the rocker switch. I was like, how is he doing that?
Magic.
Jamae, can I get a sample number?
This is sample 011.
011.
We have a viewer who would like to get a reading on the dissolved oxygen. Is that possible?
We can do that. 45.7 micromoles per liter.
Thank you.
Or is it micromoles per milliliter? I think it's micromoles per liter.
That was for you, Bobby.
It's micromoles per liter.
Yeah.
Did you say, what was the number you read out?
45.8.
Yeah, so that's micromoles per liter.
Yeah. Nice.
Awesome job, guys.
Got him.
Like I said, you guys are the best in the world.
It's just Greg and 2 other people watching it happen. But I appreciate the we. He's showing off at this point. Everybody, you can just look away. Don't give him any encouragement.
Thorough.
Yeah.
Look at
Well,
that.
it's gonna
Magic
bother me.
touch.
Tuck it in.
There you go.
That's one of those things that like you don't want to turn over. It's embarrassing.
Yeah.
Jamie,
They look
011?
Correct.
Thank you.
Yeah, the arm can be super delicate, or you can destroy things with it. Your choice, really.
Okay.
Do you want to zoom on the cut sponge, or are we done?
I think we're done.
We're done.
Let's go ahead and head on to 2E.
Okay. Let's continue on our 015 track, and then once we get up there a bit, maybe we can change over. Ready?
Ready.
Bridge, nav. One step 30 meters, bearing 015. Thanks, George.
Let's make sure we're centered up, and we're not. All right, let's come up on our delta.
What's our target?
Let's do 15. When we're stretched out like this, between like 18 and 12. Just in case. You never know.
Jamie, what was time of collection?
It's do you want the start or end or both times?
The start.
0319.
Thanks.
Wow.
What? That is
Oh man.
unsettling, huh?
You're gonna have
Gary,
dreams
do you
about
know
that.
what type of worm that was?
There's a lot of them down there. They're just hiding along the. They're
Wow.
hiding. They're blending in with the holothurian excrement. It's a good niche in life.
There you go. Well, they're cleaning it up probably.
Yeah. Yeah, look at that thing. Oh, that is the stuff of nightmares. Oh no, it's totally gonna eat a worm or something.
Or the worm's going to eat it.
Look at that thing.
Whoa.
Just, what is that?
Is that a sea
Pycnogonid?
pin?
That's a, I don't know
man.
what everybody's looking at right now, but there's a sea spider center screen. There's a bunch of worms crawling around. There were some large pink anemones. There's some bamboo coral. We got a lot going on.
The little long white, is that the sea pin or?
The long white.
It kind of reminds me of a pipe cleaner.
That's
yeah,
the bamboo
that's
coral.
the bamboo coral.
Okay.
Yeah.
Man, sea spiders are scary.
I think they're
It has
awesome.
like a growth on underneath. I wonder if it's a sponge is growing on it or what.
Gary, which one's a polychaete worm?
I'm gonna add another step.
Oh.
You go with that?
Oh, with all the legs?
Bridge now.
Oh, so these are polychaete worms.
One more step.
Yeah.
They're like just really dusty polychaetes.
Dusty polychaetes, yes. There you go, Brandy. You were interested in sediment. It's covering everything. There's so many different things that are living within this screenshot. Oh my gosh.
Whoa,
Ooh.
cool. Yeah. Yeah, look at that. Lasers are like.
You want to know what kind of fish
Okay.
that is?
That's a, I think that's a grenadier. We've got some nice lasers that went across the top of it.
It's like
So
15
nice.
centimeters
Yeah,
across, something
at
like
least.
that. Yeah, maybe 20.
And then the length of it is probably 30 to 60.
Yeah, yeah.
Yes, but it has a new name, and I can never remember the new name. What is it? Yeah.
Mushroom
What's it?
coral is
mushroom
much
corals.
better.
Heteropolypus. Thank you, Danny.
Yeah.
I'm gonna. Yes.
Better holds.
Caitlin taught me that those are dog toys because they look like dog toys.
Oh, yeah.
They look like mushrooms when the polyps are in, but they look like dog toys.
Do you say to push in?
Tentacles are up.
Is that some mat? Bacterial mat coming out of a crack. What's going on there?
Little prawn. Don't know.
Is there any flow? I don't see any flow.
No. Okay.
How's the currents down here? Right. Yeah, haven't seen that yet.
Is the ship in position yet?
We are, yeah.
Okay.
We're just heading towards 2E.
Yeah. Oh, look at that.
Oh, a little.
Little colony right on the ledge.
Yeah.
It looks like another one of those Swifties, yeah. Oh, and the polyps are all out. Hello. Lots of brittle stars in there. Gary, what's this little yellow?
Is that a single crinoid arm? What is that? Is there?
Yes, it looks like a single crinoid arm.
Oh.
Broken off.
Aww.
This looks like the colony we sampled earlier.
Yes,
Oh, it's the same exact colony?
Yeah.
Okay.
Yes, the same colony.
It's the same colony.
I could believe that. sample 009.
Do you want me to pull back out?
the newbies in the back row, we thought we were moving. We're just not.
Yeah, they kind of moved around here. Yeah. Came over, then down. Bridge nav. Add one more step. Two E.
2E. 2E.
Stop
2
there.
Edward.
There's like a million pairs up here before. Are they? You know.
Like there's a million pairs and they're both mine.
Yeah. Why don't you wear them? Is it a tunicate on a coral structure or something? It's a bag of something. I don't know. Don't know.
Could be
There's
a stock.
a tunicate right there.
Yeah, there could be a stock tunicate.
Didn't they see this on the earlier shift?
We're just.
I know they saw something very similar, and they did call it a tunicate.
Yeah, I'll just keep calling them. Change heading? Yeah. So the next one I call in, let's see, we're going, where am I going? 015 right now, 30 meters. Next one I call in will be 040.
Yeah.
So are we back over soft
035.
sediment now?
Yeah, 035.
I'm trying to look at the slope here, but yeah, I would call it soft. I would call it mixed, if you want to call it mixed, just because it's...
This is a starry night out here.
Yeah.
Mixed between soft flat and hard flat?
It's
or
it's
sloped.
It's soft on a slope.
Okay.
Cool. Look at the shadow on this Tina-4. It's just like ghost.
We have a question from one of our viewers. They said, "Oh my God, I've been waiting days and days for the underwater streams. What is the strangest things you guys have come across personally?"
Strangest. It's pretty strange. Biologically, the flamboyant squidworm.
Flamboyant.
It has to be the flamboyant squidworm.
Okay.
Where was that, Rennie?
I've
never heard that story.
Yeah.
That was cool.
Well, it's a pretty cool looking creature. You can look it up online, those watching at home. It
yeah,
shouldn't
Ed
be.
told me that was
Ed
his.
was not there. We also saw, and you can look up clips, the 2 deep staria jellies we've seen. One last year. No, sorry, one
it was
2
2 years ago.
In Revia Hagedo, and then one just this year. And then probably non-biological would be just some of the man-made stuff. We saw no trash to speak of, and then at a methane hydrate seep, a single red solo cup, just like right there next to it. It was ruining the shot.
Just hanging out.
Yeah. Bridge nav.
Saw a zebra happy birthday balloon once.
1 step 30 meters, bearing 035. Thank you. See, George listens. He's already got it in there.
Yeah.
He's just
He's spying.
He's spying on us. It's not supposed to hear all the planning, just needs to hear it.
You just wanted to hear you when you're sounding your
when
best.
I'm very confident. I don't want him to hear me waffle back and forth about it. He's listening right now.
I know. He's grinning right now, which is the best part. Little does he know we're spying back.
We heard that laugh, George.
For those listening, George is the mate on the bridge. Oh man, that Persingate is some tiny arms.
These, it looks like, yeah, they've got more prominent ones that are out in the breeze.
I think that maybe that's just had a bad day.
Yeah?
Yeah, like the ones on the left are regrowing.
Yeah, yeah.
That's the cool thing about invertebrates, though. A lot of them can regenerate pieces that are that have been lost. Sea stars are good
let's
at
push
it.
in.
Yeah, something chomped it. Man, we saw...
oh, there's a sea spider off to the left.
Where is that?
There you go.
Zoom out.
I'm zooming
Get
closer.
out of there. It's so cool, though. I think their organs are in their legs.
Are they?
There's not enough space in their body.
Some polychaetes, pictogonid.
Yeah, the little polychaete scale worms, polychaete.
Full wide.
I don't know if you really did see a scale worm or not, but there were some chitons nearby, the little scale-plated animals.
That's probably... Got a limited biological vocabulary up here.
You're doing great, Rennie. You're doing great.
Yeah, I think you guys
By
are doing
the end
awesome,
of like a coral cruise, I'll know like all the ones in the region, and then you go to another region and they're just
And they
obsolete.
look the same, but they're not the same.
Then you have to start all over.
Like this to me, I mean, is that a Rathbanaster? I don't know. Looks like that.